Okay, hello. I'm Tim Weaving, a PhD student at University College London, and I'm pleased to welcome you all to the 24th instalment in CompBioMed's e-seminar series in collaboration with the Virtual Physiological Human Institute, or VPHI. The talk today will be given by Dr Zavodszky, Assistant Professor of Multiscale Computing at the University of Amsterdam and Assistant Professor at the Budapest University of Technology. Gabor's research is focused on designing and developing detailed HPC models targeting cardiovascular diseases, and is currently coordinating the development of HemoCell, an open source cellular blood flow simulation framework that we will hear plenty more about today. Now, before we begin, I'd like to briefly remind you that the session is being recorded and will be available on the CompBioMed website and YouTube channel shortly. So please do use this to go back over parts of today's talk and others in the series, and maybe even share these e-seminars with your friends and colleagues. There will be a Q&A session at the very end of the talk, so please save your questions until then. Once the talk is finished, you can go ahead and post your questions in the questions tab, and I will read them out to Gabor to keep the session running smoothly. Now that's everything from me. I very much hope that you enjoyed today's talk, and without further ado, I will hand you over to Gabor.
Thank you very much, Tim. And let's get started. Today's talk fits, of course, in a seminar series organized by CompBioMed. And in a previous talk, Mariano did a wonderful job explaining why HPC is really important and often required component for biomedical simulations. Today, I will extend that talk a little bit by talking about why the largest scales available to us are necessary and why the movement towards exascale is important, what new challenges it poses, and what new answers it will enable us. I will use blood flow, and in particular, as Tim mentioned, HemoCell, a blood flow simulation code that I'm really familiar with, as a use case, and I will try to explain most of the things through HemoCell. That said, the overall talk is going to be structured in three different components. First, I will talk a little bit about modeling blood flow. How do we simulate blood flow in a computer typically? And then I will go a bit more into details about the numerical methods, validation, and I will mention uncertainty quantification and several other necessary components. Afterwards, I will detail much more the computational challenges which arose with HemoCell, but also I will point out how they are much more generic and typically arise with every typical biomedical code at scale. And I will also talk about ongoing research and existing solutions to these challenges. And finally, in the last part, I will cherry pick a few of the actual production runs that we did in the past couple of years, and I will show how they have relevance to clinics and experimental research as well. With that, first, I would like to show you a few snippets out of the virtual physiological human movie. This is a movie produced by the CompBioMed consortia some time ago. So if you're not familiar with this, then I really urge you to take a look at it. It's available on YouTube. All of these snippets, while they're looking quite flashy, the important part is that they are produced by validated and accurate simulations. There are multiple components showcasing the bleeding edge solutions that we can achieve today with biomedical simulations. And I think it's not a big surprise if I point out that in the lower right corner, the video regarding the red blood cells, that is actually HemoCell. And all those red blood cells that you see there, they are deforming, squeezing according to a validated mechanical model. So that is not something just for the visuals, that is an actual accurate blood flow. And to talk a little bit about the currently available scales. So if you have worked with blood flow simulations, then most likely your application fell into one of these three categories. On the largest scale that are available us, typically we are using reduced order models. That means one-dimensional, 1.5-dimensional models of the whole human vasculature network. If we are interested more about blood flows on an organ scale, for instance, in coronary diseases or, for instance, brain aneurysms, then we are zooming in much more with the volume of interest. And we have typically on the order of a centimeter or centimeter scale domains where 3D continuum models are already available for us, which can produce really accurate results. However, when we are in need for even more details, that's when we go to the right side, the cellular scales. These are really computationally expensive simulations that became available in the last, I would say, 10 years or so in small number of red blood cells. And really the recent expansion of computational power towards exascale enabled larger scale cellular simulations. This is a rough scale separation map, how you should think about the different levels of blood, depending whether your question resides on the minute and meter scale, which corresponds to the full body network, or more on the millimeter to centimeter scale, where the 3D continuum models are available, or you need to have information from scales below that level. And of course, as you can see on this figure, the computational cost naturally grows with the increase of the detailed information. Now, some of the questions reside on levels where, for instance, the 3D continuum model is more than enough. But with the progress of current clinical practice and clinical research, more and more of the biological and chemical processes are included in these simulations. And for those, more often than not, the cellular and subcellular level is necessary. I will give some pointers for this, and I will show why and how. But first, let me go into introducing the most used code, the spotlight code of this talk, HemoCell. In the background, again, you see a part of a blood flow. However, the deformation and flow of the red blood cells are according to validated mechanical models. So again, this is a flow that is actually accurate and realistic. The code itself has been fully validated and went through detailed validation, verification, and uncertainty quantification. One of the major points of HemoCell is that it is optimized for high-performance executions. That is, it has a demonstrated efficiency over 300,000 cores. It also provides various advanced boundary conditions that are necessary to match experimental settings and to match clinical settings. And it can maintain numerically stably a sustained high shear rate. That is important because many of the pathologies appearing in the human vasculature, they have to do with really high shear rates. That is thrombus formation, stroke, embolism, ischemic problems. These are all related at some point to very high levels of shear rate. So that is necessary that we are able to model this. Apart from this, we also have various cell types and several detailed internal structures for the red blood cells. I will mention the important ones. Now, for this talk, one of the important points is that HemoCell itself is a coupled code, meaning that we do have a continuum 3D simulation that computes the blood plasma underlying, and then we have the cells that are immersed in this fluid. This has consequences for the computational techniques that we are using, and I think this is a good example because many of the codes and simulations that target bleeding edge problems are coupled simulations. Often we have some kind of a flow like respiratory issues, you have airflows, and solid parts like the lung itself coupled to that flow. So I will use this opportunity to mention at several points during this talk why coupled simulations are different and what challenges they pose. As a starting point, recently at a conference, we made a survey about the importance of various components of numerical codes. And one of the top importance for most of the users turned out to be the access to validation data and documentation. So therefore, I thought that I will just quickly point out that HemoCell is a validated code. We have standard validations for single cells, that is cell stretching and cell shearing, for instance, comparing it to experiments. And we also have validation for many cell flows where we are flowing blood through larger channels. And then we look at the overall viscosity and other bulk properties like cell-free layer. This is for HemoCell, but for upcoming medical and biomedical computational applications, it is of utmost importance that the validation is not just done in terms of comparing fixed parametric sets to experiments, but also to carry out larger scale sensitivity analysis and uncertainty quantification. In this specific case, the sensitivity analysis was also useful to reduce the number of parameters that we have. For a given use context, we carried out this sensitivity analysis to look at the different parameters and how they influence the outcome of the simulation. What you can see on these three figures is that the first one, this parameter that defines the young modulus, that is the stiffness of the cell membrane, has an almost 1.0 effect on the overall outcome, meaning that it's a really, really important parameter. While another one, which defines the bending stiffness, the bending parameters of the cell membrane, is closer to 0, meaning that it has very little effect on the outcome. These sensitivity analyses are quite important because they define which are the important parameters and which are the parameters that we need to take into consideration very, very accurately, and which are the parameters where a bit larger uncertainty might be allowed. With that, we also took several cell types, not just healthy red blood cells, but diabetic, malaria, and different other types for uncertainty quantification as well, to figure out that all these parameters and uncertainties we have on the parameters, how do they influence the final uncertainty of the output? This type of investigations are computationally can be really expensive. So most numerical codes, what they do is that they create an intermediate simulation, which is usually a surrogate model, for instance, based on machine learning or some other neural network technique, that can reproduce parts of the full simulation and can be sampled really efficiently. The important part is that uncertainty quantification in itself is really computationally expensive and can make a requirement for HPC execution, even if the original code was smaller scale. Now for HemoCell, of course, the original code was already a large scale code, so this topped up the requirements a bit. And to look at why the computational cost is high. Well, previously, this kind of unprecedented detail was just not available, because if you look at a single red blood cell, it's a really complicated structure. It has hemoglobin inside in order to be able to transport oxygen. Hemoglobin is a highly viscous fluid, and of course this whole structure is optimized by evolution to be able to pump blood through with this high viscosity hemoglobin content with the least amount of effort, so that your heart has to spend the least amount of effort on pumping these blood cells through the whole vasculature. But in order to be able to do this, the red blood cell has to respond to various mechanical requirements. And inside that hemoglobin, it's surrounded by a cytoskeletal structure, and it's also surrounded by a membrane that can detach from this cytoskeleton and can rotate around. You can see on the right side a bunch of red blood cells randomly pulled out of a detailed simulation, just to show all kinds of mechanical deformations these cells undergo in a simple flow. You can see that they are sheared, they are squeezed, the outside membrane can actually rotate around the internal structure, and all this needs to be modeled in order to get a really accurate picture on how the cells behave in flow. This is one side of the story. The modeling of a cell itself is quite expensive. The other side is that if you just look at a tiny, tiny drop of blood, let's say 1 microliter, that's not even a whole drop, that's just a part of a drop, it does contain in the order of millions of red blood cells. So even to reproduce a very tiny part of the human circulation, we need an enormous amount of these red blood cells, and these red blood cells need to be able to capture tiny details of deformation. All this together, of course, leads to really high computational costs. This computational power was not available until recently, but it's not purely that we have now bigger computers, so we can just take our simulations and run it in a bigger size with weak scaling or a longer time for strong scaling. There are, of course, additional challenges that arise with these scales. And on this scale map, I showed a number, of course. It's just an order of magnitude estimate. And I would say that up to the order of thousands of cores, we have everything figured out already. Starting from tens of thousands of cores and reaching into the first 100,000 or so, There are new challenges on the horizon. Typically, they have to do with the software stack available on the HPC computers. There can be numerical overflows, all kinds of errors, which require now new standards, for instance, the upcoming MPI-4 standard. There are additional challenges with porting the code itself, since most of the new HPC architectures that are becoming available, for instance, in Europe, much of the computational power is provided by accelerator cards, GPUs. That means that the current codes need to be transported, need to be ported towards this GPU execution, so that the codes have either a pure GPU or a hybrid execution model, where part of the computation is done on the CPUs and part of it is on the GPUs. Above these, there is a bottleneck of the input-output operations, because we need to write out files. And of course, large simulations also mean large output. These challenges are relatively small, approaching exascale. However, when our simulations reach the 100,000 cores, and we have experience with this firsthand with HemoCell, then some of these problems become really severe, and they require new solutions. For instance, load balancing in order to improve performance, both static at the beginning of the simulation and both dynamic during the simulation run. Energy efficiency is becoming an important factor, and I will detail a bit later what is the energy cost of an application like this. Also, resilient computing and fault tolerance becomes important, since if we have a really large simulation that runs for days, if there is a problem at CPU 200,008 and the simulation fails, we need to be able to recover it in order to avoid running the whole thing all over again. Then for codes that are smaller or have a different execution pattern, for instance, molecular dynamics codes, which are typically one simulation is not large, but because we need a large parametric space, we need to run a swarm of simulations together, and that's how we use exascale. There are additional challenges with the management of complex workflows. Currently, we are drawing data and experience primarily from these HPC centers. HemoCell is deployed in all of them, and we have support in terms of script and documentation on how to execute code on all of these centers. As for the actual decomposition, how it looks like, in this video you can see a short flow of cellular blood flow. Every single color-coded block is a different core. So at the beginning of the simulation, the whole domain is decomposed into these different blocks and assigned to different CPUs that are computing these blocks. But this is static in terms of the fluid. However, if you look at the cells, they are obviously moving compared to this flow field. So they need to be handed over to different cores, and this creates a quite complicated communication pattern during the run. The overall decomposition always happens in these colorful rectangular domains, and the communication itself is implemented in MPI, as with basically all large-scale simulations, as this is the de facto standard. One important challenge here is this passing on of the cells, because there are other codes which also assign the solid part, the cells, to the same cores. However, in order to enable efficient communication between the cells and the fluid, we need to keep computing every single red blood cell at the core where the fluid field where it currently resides is also computed. Otherwise, we would introduce communication that is not local between the fluid and the red blood cells. A small trick that we do in order to speed up the beginning of the simulation. This is something that typically does not arise as a problem unless somebody actually wants to try to simulate cellular flows. It's how do we initialize the cells at the very beginning? And for that, we employ a separate simulation, which is also parallel, that computes the position, the initial position of these cells. And when we start to run these simulations at really large scale, this seems like a quite simple slide, but it is a quite important one because it talks about money. On the left side, you can see the overall computational efficiency of a really large scale simulation as we go up with the number of cores. The last data point that you see corresponds to 330,000 cores. And I put a red line at 1 million core. That is, let's say, that is the entrance part of the exascale region. So you can see that currently with our capabilities, both in hardware and software, we are really close to this red dashed line. And the fact that we can maintain above 70% computational efficiency in a coupled code where red blood cells are moving and deforming and doing all kinds of things, I think this is remarkable at this point. However, if we start to talk about the cost of such a simulation and how much energy it uses, then please take a look at the right side. That is an extrapolated figure. It uses the same number of CPU measurement points as the left side. However, while on the left, we just had a shorter simulation run in the order of 10-30 minutes just for benchmarking purposes. On the right side, we extrapolate this to actual production runs, which are typically up to 1 second of computed flow. That corresponds to, let's say, 2 days of HPC wall runtime. So these dots are large-scale simulations run for 2 days. And on the y-axis, you can see the energy cost in euros. And what you can notice is that for the largest scale run, which is getting close to exascale, we already reach in the order of 100,000 euros for a single simulation. That means that that one simulation probably uses more energy than my whole household will in my whole lifetime. So these simulations are super costly. They enable an unprecedented level of detail where we can ask much more questions than ever before. However, they are super expensive. And for that reason, we need to address the computational challenges with much more accuracy than previously, because it's just super expensive to waste energy, to waste core cycles. And in order to do this, there are many strategies that we follow, and I will outline some of them that typically also applies to other coupled codes. One possibility in order to reduce performance cost, to increase performance basically, is to look at the different coupled components and their timescales. In this particular figure, I show a dynamic type step size, which is based on watching carefully what happens in the simulation. The blue line on the left is technically the arising forces. So we are looking at the blood flow. There are collisions of cells and deformations, and we are looking at how violent, how extreme they are. And while the whole flow is peaceful, not so much deformations, not so much aggressive collisions are happening, we can afford to take much larger time steps and make the simulation much more cheap. And when we approach around 800,000 or so a region where more events are happening and the forces in the system start to accumulate, then we can scale back the time step size, go for a smaller one, and go for a faster simulation. Another typical problem that will arise at every single simulation, not just blood flows, but everywhere where we have coupled components that move compared to each other, is the appearance of load imbalance. Static load imbalance is something that's long been known, that there is a demonstration in the top figure where you can see a cylindrical vessel flow and the domain decomposition. And what you can notice is there are colorful blocks at the corners, which are basically empty. This is just a regular decomposition, most naive one possible, but it leads to load imbalance because there are blocks assigned to CPUs that are not doing anything since those blocks are empty. This is something that is relatively easy to solve at the beginning of the simulation. The more challenging part is dynamic load imbalance. That happens due to the movement of the cells in this case, but in every other coupled simulations, if you think about the respiratory system, while the lung moves, it changes compared to the fluid domain. So the way at the beginning we distribute load across CPUs might not be valid anymore. And this is a really important part, and it can introduce a huge overhead of computation. If you look at the right side, that is the typical simulation run for a couple seconds of physical time, where we measure these overheads introduced by load imbalance. There are different types of imbalances. For now, let's just look at the green curve, which is a computational imbalance. It keeps growing, and when it reaches 1.0, if it would reach 1.0, that would mean that the overhead is exactly as large as the computational cost without overhead. So we are taking twice as many computational power to compute the same thing. So therefore, we need methods in order to be able to capture this and do something about this. We should be able to notice these overheads and then redistribute the computation during the run. That happens at the red vertical line that you can see in the figure. We stop the simulation, redistribute everything according to the current state of the red blood cells, and then we restart it from a much more balanced situation. How this looks like in practice. There is a very simplified Y-shaped bifurcation on the right side, but it is enough for demonstrative purposes. You can see that the red blood cells are moving through. And another thing that you can notice is that the domain decomposition, denoted by these colorful blocks, they keep changing. In this particular case, the solution is that we can compute the cost of every single, the computational cost of every single of these colorful blocks. And when some of the blocks is next to each other are very cheap, then we just merge them. So you can see the appearance of these larger rectangular blocks every now and then when they don't contain many cells. And at parts where we have many red blood cells, we can again split up these larger rectangular blocks to smaller ones and assign them to different CPUs. There is one caveat with this technique, and that is, how do we know when to do this? Because for such a small simulation, it's a relatively easy task, but as you can imagine, if we go to really large simulations, stopping the whole simulation, saving the state, redistributing everything and starting up the simulation again has, of course, an additional overhead. So for these reasons, it's getting more and more important to develop performance and also energy prediction models. We are doing this for HemoCell, and the primary aim is that these performance models can inform us that if we are stopping the simulation right now and would do a rebalancing, how much performance benefit would we gain? And at some point, that is larger than the cost of stopping the simulation, and at that point we can trigger this redistribution. But in order to be able to do that, we need to be able to model really accurately how the performance will change and also how the energy consumption will change, since in many of the future applications, the computational budget will not going to be core hours anymore, but probably some sort of an energy budget. So we need to be able to tell our simulations when to checkpoint, when to rebalance in order to get to the lowest energy cost possible. That is eventually the cheapest simulation possible. And of course, there are additional important components next to how to achieve the best performance of the lowest energy cost. It's also important for many of the codes, for all of the codes within the CompBioMed portfolio, to develop user-facing resources, including documentations and example cases. And in the same study I mentioned, same query I mentioned previously, where the most important part was the availability of validation data, the second most important for the users were the available of documentation, tutorials, and other user resources. So for most of our codes, you can find, of course, documentations, examples, tutorials. For HemoCell, we also have recorded terminal sessions to provide a step-by-step instruction how to deploy this code on 2 different HPC centers. We also have prepared module scripts for many of the HPC centers in Europe that describe how to deploy this code on them. And currently in the works are various deployment tools that will make the deployment of the codes much easier. Apart from this, you can also find various videos and additional information on the CompBioMed YouTube channel. And with that, we get to one of the last challenges that these really large-scale codes provide. And that is, how do we visualize and how do we process the data coming out of these super large simulations? And as you can think, as you can imagine, when we have millions of red blood cells, where we have detailed information on the forces on them, on the deformation, on their position, that is in itself a huge data set. And therefore, I think it's also important to mention that all of these really large simulations come with capabilities on high performance data analytics and visualizations that are also HPC applications in itself. So it's not just that we need HPC and really high performance for running these simulations. Oftentimes, we also need it in order to be able to set up these simulations, provide initial conditions, and at the other end of the simulation to visualize it and to post-process the data. What you can see on the right side video is a peer-review session visualizing a large-scale simulation of red blood cell, where again, every single colored block is a different core. So in order just to visualize this data set, we had to use 128 different nodes. This really is a large-scale simulation. With these challenges and with these capabilities that I've shown, I would now transfer more to how are these applied and what are the typical production applications at the moment. And then I will also tell a little bit about how these applications will probably evolve and how they will look like in the near future. First off, HemoCell is applied to various vascular diseases. The main focus currently are for malaria, diabetic diseases linked to various retinal problems like diabetic retinopathy, then all kinds of thrombus formation diseases that can happen both in arterial and in venous circulation. And then finally, we have a focus on cell and particle transport at the moment, but we are moving towards adding more and more chemical and biological processes in order to model even more complicated biomedical processes. One of the, or some of the common points of these diseases, while they were not really possible, let's say 10 years ago, is that they require sustained high shear rate. That is typically something that happens with thrombus formation. They require advanced boundary conditions, and they are large-scale simulations. That is in the order of at least tens of thousands of red blood cells. One example of this was recently done by one of our PhDs, looking at what are the initial conditions of high-shear thrombus formation. High-shear thrombus formation causes a lot of problems in human vasculature. It can lead to heart or brain infarcts when some of these thrombi break away. And they typically develop around vascular stenosis, so narrowings at the human vasculature, or around micromedical devices that can be implanted, stents, for instance. The question here was, all right, what are the typical conditions and the typical location when we have a stenosis, like this mock stenosis that you see on the top? Where will the thrombus form? And of course, we can do experiments first, and that's what you see in the video, one of the blood flow chamber experiments. The problematic part is that blood is not transparent, so we cannot see what happens inside, and it's really difficult to measure because if we put any kind of measurement device there, that will disturb the blood flow. It is even problematic in an in vitro situation, and of course in an actual living patient, in an in vivo situation, it's next to impossible. And that's where blood flow simulations are coming into place, because we can reproduce much of this flow in high detail. That's some simulations you can see on the lower right corner. And these simulations can inform us what happens inside the blood. What are the different components doing, how they are transported, and where you can find them, where are the high availability points. And we learned quite a lot from these simulations. Now we can predict the actual position where a thrombus will start to form. We know what are the conditions necessary for this formation. And that has severe implications for various thrombus formation and bleeding related disorders. An additional simulation and project that we've been doing is with diabetic diseases. Diabetes is a widespread disease, and there is a lot that we don't understand about it at the moment. But one thing that we know is that diabetic red blood cells have different mechanics. They are more stiff. they undergo various chemical oxidative processes that makes them stiff. So we have now implemented in HemoCell multiple cell types, diabetic cell types, that you can see 3 different cell types in the right side. The red ones are healthy red blood cells. They deform easily when you put them in shear flow. That's a normal flow in a vessel. You will see that they flatten out, they squeeze, they deform. However, diabetic cells, depending on their status, are more rigid. That's, for instance, the green and the blue one. The green has some deformative capabilities, and the blue one is completely rigid. We have validated these cell types with diabetic cell types, and they underwent the same VVUQ process as the healthy cells. And using the simulation was again necessary because even though we could do experiments both in patients and in microfluidic chips, since blood is not transparent, we can only tell what happens with the cell in the closest 10 micron or so to the vessel wall or to the microfluidic chamber. We cannot see deeper in the flow. So what we do with the simulations, again, we match the conditions of the flow. We simulate exact same flow that we have in the experiment as well. And we make sure that whatever we can see in the experiment matches completely what we find in the simulations, but then the simulations can give even more information. And they can tell us, for instance, in this case, how platelets, an important small component for hemostatic processes, how they are transported in the different cases. And we found that in a diabetic situation, when the red blood cells are stiff, then the healthy transport of platelets are disrupted completely. They don't get next to the vessel wall where they should be. So in a diabetic case, if you have some injury or cut, then the prevention of bleeding, the sticking of platelets that cover the hole or the wound area, that happens much, much slower. You will bleed much longer. This is something that was known, but the mechanism was not known behind it. And similarly, again, it's a diabetic situation. Another issue that arises with diabetes is that in the eye, the human retina, these small lesions will start to appear. And that is problematic because when they rupture, that's a bleeding in the eye, and that is a leading cause for blindness in many of the developed countries, this diabetic retinopathy. So again, we could use detailed experimental measurements on the shape of the retinal vasculature, and then use detailed numerical simulations in order to look at how the cells are transported and what type of forces they exert on the vessel wall. And eventually we found that the formation of these small aneurysms are most likely linked with this stiffened state of red blood cells because they cannot squeeze through the tiny vessels of the retina easy enough. And they exert a mechanical load on the vessel wall, a constant scrubbing, if you will, which eventually leads to inflammatory processes and the development of these small aneurysms in the retina. And finally, the last example that I would like to show is what we can learn. It's a currently ongoing project. We are trying to learn about how vessels and blood flow behaves in terms of a puncture. Now, we are looking at scales of puncture from microneedle, or you can also look at the mosquito on the left. The needle of the mosquito is in the same order of magnitude. And we create a blood flow model where at the side of the vessel we have a circular puncture, and we look at how the flow behaves while flowing through this wound, and also how the flow behaves downstream. And this is a larger scale simulation. It's not one of the largest that we've been doing, but this is already a simulation that requires in the order of tens of thousands of cores to do efficiently. And you can see how the blood is flowing in from the left side, and then some of the blood passes through the circular wound on the top. And we can learn a lot about this process that is not possible with either continuum flow simulations or with just pure experiments. In the experiments, we can measure how long it takes for a wound to stop bleeding, for instance. From the simulations, we can tell the availability of platelets, which explains why the wound takes that long to get to hemostasis fully. And additional things we learn is that downstream, the hematocrit changes, the availability of various cells changes. So if you have a punctured wound, that influences behavior of blood and behavior of various processes downstream as well. With that, I would like to spend a few words on, all right, this is the current state of the art. Where are we going with this? Well, the current developments are mostly focusing the incorporation of various biological processes. That is, for instance, we are looking at thrombus formation, which is a really complicated cascade process with various components and various interlinked processes. This is becoming possible with the computational scales that are currently becoming available. So these questions, they reside on a clinical time scale of seconds, minutes. However, the components that played a major role in them, they are microscale processes. So they are on the level of platelets, of smaller proteins, and on the time scale of microseconds. And in order to bridge this scale, these scales, we need to do various things. On the one hand, we introduce multiple subcellular processes like uncoiling of von Willebrand and other protein behaviors in the cellular simulations that require really high computational power already. But this in itself is not enough to get to the actual organ scale simulations. What we also need to do is introduce multi-scale modeling concepts where we have a larger scale simulation that represents the whole organ scale if it's necessary. And it is informed by smaller microscale models, a swarm of them, that can compute the really micron scale processes as they are becoming available for these cellular simulations. And these are not things of the future. This is the current development. So my prediction would be that in the upcoming 2 or 3 years, more and more models will be able to provide capabilities that are strongly multi-scale and can incorporate from the smallest protein level to the organ scale simulation levels, and they will open new doors for clinical practice and therapeutic practice because they will be able to inform us on various details of a specific drug, for instance, of specific patient-specific details. And this is something that is currently in development, so this is not sci-fi, this is happening at the moment. But in order to make this work, we need the big computers, we need the access to exascale. And with that, I would like to thank many of the partners who contributed this work, and I would like to give the word back to Tim. Thank you.
That's fantastic. Thank you very much, Gabor. I very much enjoyed your talk. So just a quick reminder for the audience that if you do have any questions, now's the time to go ahead and submit them. So you should be able to see a questions tab somewhere on your screen, and if you go in there, you'll be able to submit your questions so that I can read them out for you to Gabor. Okay, and so whilst you're all gathering your thoughts, I've got a few questions of my own for you, Gabor, if that's okay. So just at the end of your talk there, you were talking about how we might couple these microscale simulations with, say, larger systems biology codes on macroscopic scales. So, you know, what are the main challenges associated there, really, to achieving that that coupling? To, you know, this is all in the striving towards, say, virtual humans, digital twins, etc. You know, what are the challenges involved there?
Yes, clear. Thanks. It's a very good question, and it's also a very difficult one because we have multiple challenges. So far, computational capability was, of course, one of those. With the upcoming computers that becoming available, for instance, Lumi or Leonardo, or if you look at the state's frontiers, this is less and less of a challenge. However, there are many scientific challenges on the biology part. Those are typically related to gradients on the smaller scales, and how do you propagate that information towards the larger scales. And for now, it seems like that the solution in most cases will be the introduction of some sort of a statistically correct model that is informed by the microscale models. However, it cannot represent all the details of the microscale models, but there is a statistically correct coupling between them that can inform the macro scale. This is, I would say this is quite bleeding edge, but I see this happening over various different biomedical codes. And of course, we are also doing it with HemoCell.
Of course, perfect. Thank you. This is something I'm very interested in myself. Being a researcher in quantum computing, I'm very interested in how these microscopic sort of scales can traverse these different these multi-physics, multi-scale problems. So that's something that I'm very interested in as well. So talking about the sort of computational hurdles that need to be overcome, so to speak. What about, say, GPU acceleration? You know, how optimized already is HemoCell for use with accelerators, and might there be additional performance that we could still that is yet to come, perhaps?
Yes, that is a somewhat painful point. You pinpointed it quite well. At the moment, HemoCell is CPU only, but we have multiple efforts to move it towards GPU, even more so because, as I've mentioned, it is a coupled code. So there is a component of fluid, which is a fully structured grid, and it's very well suited to be ported for GPUs. The other component, the cells flowing in the fluid, they are, of course, represent an unstructured grid and maybe a little bit less suited. But overall, we are planning a hybrid execution where the cells are executed on the CPUs and the blood plasma, basically the fluid field, executed on GPUs.
Right, I understand. Okay. Right, I'm not seeing any questions from the audience yet, so I'll continue with a few of my own if nothing else comes through. So within the examples that you showed there, we've seen a few different domains, right, that have been specified, and then we've looked at, say, the blood flow with respect to the domain. Does the complexity of that domain have an impact on the performance, you know, through perhaps more time-consuming load balancing or anything of that sort of nature?
In specific, I think that cellular blood flows are one of the leading examples why we need load balancing, while load imbalance does happen with almost all coupled codes. For specifically cellular blood flows, because red blood cells tend to move away and they have various properties, like they like to stick together under certain conditions. It is a really good example where the load imbalance can easily get up to 100%, meaning that the overall computational cost is now doubled just because load imbalance overheads. So this is a prime example where we really need good solutions in order to tackle these questions. Now, depending, of course, on the question at hand, it also changes. With simpler geometries, we can have a much better initial guess on the domain decomposition and on the computational cost. With much more complicated geometries, for instance, vascular networks, larger networks, this question becomes harder and harder. And while we already do have various load imbalance solutions, one of the leading ones are developed, for instance, at Barcelona. The question of dynamic load imbalance that happens during the run, so cannot be predicted at the beginning of the simulation, is still very much a bleeding edge research area. And I hope that we will see new results also in terms of energy efficiency, there where the similar models and load imbalance costs are applied not just to wall time and core hours, but to energy usage of the simulation, which actually can differ under some circumstances quite a lot from the wall time. So that's a completely different optimization target.
Sure, absolutely. Well, thank you very much for answering my questions. Those were the ones that I had. I haven't seen anything else come through from the audience, so unless something comes through in the next maybe minute, I think that maybe this would be a good point to start to draw the session to a close. So thank you once again, Gabor, for giving us your time today. Just a quick reminder to the audience that this e-seminar will be available on the CompBioMed YouTube channel and website shortly, which is accessible on the link on the next screen. If you don't mind flipping over for me, Gabor, that's the one right there. Now, before we end, I'm just going to take a moment to plug CompBioMed's free scalability service, which is on the next slide, please, Gabor. So if you are someone who uses or even develops biomedical applications that necessitate intensive computation, but maybe you're running into performance issues that are limiting the scope of your research, then CompBioMed offers a service completely free of charge to improve the turnaround of your computing jobs. Next slide, please, Gabor. So if this is something that applies to you and your research, you can fill out an application form available on the web page displayed at the top of the screen. Otherwise, please feel free to reach out either at the email address shown here or via the In Silico World Slack channel. Next slide, please. So In Silico World is a community devoted exclusively to in silico medicine, offering expertise, opportunity, and collaboration, and a safe space to exchange ideas and advice. Next slide, please. Already, In Silico World has a large following, including members from large biomedical companies, biomedical SMEs, independent software vendors, clinical research institutions, and even regulatory and standardization bodies such as the FDA and NICE. So finally, thank you to everyone in the audience for attending. Thank you once again, Gabor, for that fantastic talk. I really enjoyed learning about those different use cases that you went into there.
Thank you.
Thank you again. So I hope that you all enjoy the rest of your day.